We know with adults, stress tends to be a component, more so than sexual hormones, which are responsible for teen acne or the acne associated during puberty. We know that in adults, because of the aging process, our skin tends to heal much slower. That redness lingers longer with an acneic blemish. We know that with adults, we tend to have combination skin. You might have alipoid or dehydrated skin at the cheeks and still an oily T-zone or breakouts on the chin and the jawline, which are very commonly associated with the adult acne condition. We know with adults that there's an increase in sensitivity, and because the stress factor, cortisol hormones are secreted from the adrenal glands, and this makes pigmentation more prevalent in an adult acne client than the teen client. So all of these factors, these differences between the adult acneic client and the teen client have to be taken into account when developing a protocol and a treatment regimen to treat your client. Before we can understand how the Medibac system has been designed for treating the adult acne client, let's review what we know about the acneic condition from a histology and physiology point of view, as well as how certain ingredients can impact that. Let's look at our model here to talk about the pathology associated with the acneic skin condition. Acne is a disease of the pilosebaceous unit, and that's namely the hair follicle here. It also involves the sebaceous follicle. A sebaceous follicle looks very similar to the hair follicle here, only the sebaceous gland, which is right here, is much larger. It would take up a much vast space as opposed to the hair follicle here. And the hair in a sebaceous follicle is much finer. We refer to this as a vellus hair, and sometimes it doesn't exist at all. Now what's happening, whether it's the hair follicle or a sebaceous follicle that is involved, is the sebaceous gland gets stimulated to secrete sebum. In the case of the teen client, what's happening is testosterone, which is secreted from the testes in the male and the ovaries and adrenal glands in the female, goes through the blood system and enters into the sebaceous gland. There it's acted upon by an enzyme, 5-alpha reductase, which converts testosterone to dihydrotestosterone. This immediately turns the green light on to sebum production, and sebum is then not only produced but secreted into the canal of the follicle. It can be the hair follicle or the sebaceous follicle. The sebum, if there's a hair present, literally wicks along the hair and goes out to the surface of the skin, and you can readily wash that away. Now, in the case of adult acne, where stress is really the trigger, what we find is that the adrenal glands, when we're stressed, secrete testosterone and secrete epinephrine as well as cortisol, and these can also stimulate glandular activity to increase sebum production. When sebum production increases, we find several things are happening. We find that when sebum production increases, we alter the calcium gradient. There are calcium ions throughout this epidermis. And when sebum gets to be in excess, we alter the calcium gradient. And what scientists have just discovered in the last few years is when we alter this calcium gradient, we stimulate proliferation of cells. Now look here, this is our epidermis here, and this dark blue line is the stratum germinativum. And notice how it is invaginating down into the follicle. So what you're finding is this stratum germinativum, which is responsible for cellular division, giving rise to daughter cells that move on up out through the epidermis during keratinization, this is still occurring in the follicle as well. Well, once we alter this calcium gradient, what we've done is we've caused a proliferation of cells, and lo and behold, they tend to accumulate in the neck here. We also know in the acneic client there are less lamellar granules in the stratum granulosum layer, which is just underneath the stratum corneum. What does this mean? Well, the lamellar granules are responsible for secreting lipids, which comprise the barrier lipid layer, but they're also responsible for secreting the enzymes that are responsible for exfoliation or desquamation. What does this mean if we have less lamellar granules? It means that we're not going to be sloughing and exfoliating off the cells. So what we end up with is a lot of cells proliferating here, and we're reducing the exfoliation on the surface of the skin and even in the follicle. So you take this excess sebum combined with this proliferation of cells, And we find we get this soupy, gooey mess in here. And what basically happens is we form an impaction plug. Think, if you will, of putting a cork inside of a wine bottle. This impaction plug is doing the same thing to the follicle. It's clogging it. It's stopping any release of the sebum from going out to the surface. It's trapping it down in the follicle. So now we have this impaction plug, excess cells due to the proliferation in the neck here of the follicle. We have excess sebum, and we have bacteria that normally reside. In this particular case of acne, it's Propionibacterium acnes, otherwise known as Corynebacterium. Propionibacterium acne loves to eat off of the sebum, and we now know it has an enzyme called a lipase, and that lipase digests the sebum, releasing fatty acids into the follicle here. Those fatty acids, which are rich in double bonds, are prime targets for free radicals. The free radicals break down and attack those fatty acids, and that's where we get the subsequent inflammation occurring in the follicle. We also know that in the acneic skin condition, we have an increased permeability, actually a dysfunctional permeability layer in the lining of the follicle. And so we're able to have inflammatory modulators and movement of things throughout this follicle into the surrounding tissue, which can lead to inflammatory pustules in this region here. So our knowledge of what's happening in the actual skin histology and physiology is much more extensive today than it was even a few years ago.